Bismillahirrahmanirrahim. Assalamu alaikum everyone. Hope you guys are doing well. We are the researchers from the Department of Microbiology and Molecular Genetics, the Women University Multan. My name is Manahil Khanam Durrani and I am here to present our group project, which is based upon physical and microbiological characterization of treated and untreated sewage water. And my group members include Manahil Khalid, Manahil Khana, Anika Rana, Aisha Khalid, and Chanjalati. So first of all, let me tell you that what is wastewater and why there was a need to do this group project. So water is one of the basic requirements of human daily consumption. Yet about 2 billion people in the world live without access to safe drinking water. Pathogenic bacteria which causes diseases such as typhoid fever, dysentery, diarrhea, cholera is indirectly studied by studying indicator organisms, and the most common indicator organisms are coliforms. So the 3 basic tests to detect coliform bacteria are what presumptive test, confirmed test, and completed test. So the present research is focused on performing different lab scheduled tests for effective treatment of domestic wastewater from Sikandari Nala Multan, which was our source from where our sample was collected, and this wastewater ultimately pollutes the environment. So our first step of the group project was sample collection. We collected our sample from Sikandari Nala Multan. So our first step, sample collection and water treatment, will be explained by Ms. Anika Sani.
I will tell you about the sample collection. As you can clearly see from the picture, that sample was collected from the Sikandari Nala Multan, Pakistan. We have preferred this location as it is present where industries discharge their waste. We have visited that location and collected our sample from there and moved to our lab where further processing was done. First of all, there was a physical characterization of sample, and we have analyzed that water was greenish black, having poor smell, and its pH was recorded as A. Over physical characterization was the first step of water processing. For this purpose, we use sand filtration as well as UV and filter paper technique. Sand filters are used as a step in water treatment. Our sand filter was consist of a plastic bottle, 6 cm of gravel was installed at the bottom of the bottle, and we put 6 cm of sand and 4 cm of charcoal and covered it with bandages. After sand filtration, we have done filtration with filter paper that had pore size of 0.4 micrometer. It was further processed with the UV exposure of 30 minutes. Then we observed physical changes in water, as after the treatment pH was intact, clear, smell was slightly reduced, and we observed a huge difference between the processing after the processing done. We had done qualitative and quantitative analysis of both treated as well as untreated water, which will further explained by our group member Aisha Khalid.
After the treatment of water, we got 2 samples, treated and untreated, and 2 samples were analyzed side by side by different tests. First of all, we performed qualitative and quantitative analysis of coliforms. Coliforms are basically gram-negative, non-spore-forming bacilli that possesses beta-galactosidase that produce acids and gases. Qualitative analysis consists of 3 tests: presumptive test, confirmed test, and completed test. In presumptive test, different types of dilutions. In first 5, they were 2x, and they contained 10 ml of sample in broth, lactose broth, and then 1 ml of sample in next 5 tubes of broth, and 0.1 ml in the next 5 broth tubes. And when we compare treated and untreated water, in untreated water, all the tubes contain bubble, and the amount is 555, and its ambient is most probable number is 2400. And in untreated sewage water, all the tubes contain bubble except the last one tube, and its value is 554, and most probable number is 1600. The next one is confirmed test. In confirmed test, we take inoculum from the lactose broth and treat on EMB plate. And after the incubation, and we took the colonies from the EMB plate and treat on the NIG. Next come quantitative analysis of coliforms. It consists of two parts: total coliform test and fecal coliform test. In total coliform, we use the media violet bile red agar, and for fecal coliform test, we use the media EMB. And for both types of media, firstly we diluted the samples up to 10⁻⁹, and through these dilutions we poured our plates for total coliform and fecal coliform, and then after spreading, we incubate it, and after we get colonies, we isolate the colonies through cross-street or quadrant street method. And as you can see the results in quantitative analysis, in untreated water, there were 30 colonies in total coliform count and 22 colonies in fecal coliform count. While in 10⁻⁹ dilutions, there were 7 colonies in total coliform count and 10 colonies in fecal coliform count. In treated water, there were 12 colonies. Water at 10⁻⁵ dilution, there were 12 colonies and 14 colonies for fecal coliform count. While at 10⁻⁹ dilution, there were 2 colonies for total coliform count and 4 colonies for fecal coliform count. And in comparison, there were more number of colonies in untreated water as compared to treated water. After obtaining different strains from quadrant streaking, we have to confirm these strains through biochemical tests to characterize whether it is which type of bacteria or it belongs to which type of species. And this will explain by Shazal.
I will explain the biochemical analysis of the coliform. These biochemical analysis, they are used to confirm the isolates that we have claimed. Either they are the coliforms, means they are gram-negative organisms, or the gram-positive organisms. So in biochemical test, we have performed IMBIC test. IMBIC test, it is a combination of 4 tests. It involves indole test, methyl, Vox-Prosker, and citrate utilization test. For different tests, we have used different media culture. Like for indole test, we have used sim agar. For methyl red, we use methyl red agar, and Vox-Prosker also involves the same methyl red agar. While for citrate utilization test, we have used citrate. So in the indole test, what happened is that after inoculating the culture, 24-hour old culture, incubating for 24 hours, we have obtained that this after addition of Covax reagent, 10 drops of Covax reagent, a deep red layer formed, which shows this species, the formation of the deep red layer, that shows that it is a species E. coli is formed in this particular test confirms the presence of E. coli. As we have seen this in the biochemical test of wastewater, we have 2 water samples, treated or untreated. In untreated water sample, the strain we have obtained is E. coli, and this strain, we have confirmed this strain by Burgess manual. That shows that E. coli will always give indole positive test, methyl red positive, Vox-Prosker negative, and citrate negative test. Means that methyl red is confirmed by the formation of red layer, cherry red layer. Vox-Prosker is also formed by the formation of red layers, and citrate utilization test means that the citrate green color is turned into blue color. Means that this E. coli has utilized citrate as a carbon source or energy source. The same is the case with Pseudomonas species and Klebsiella species. And these species, Pseudomonas and Klebsiella, but the test they have, the confirmatory test they have given to us, is further confirmed by Burgess manual. While in treated water, the biochemical test analysis have shown that in treated water there is no Pseudomonas species, only E. coli and Klebsiella species. And these Klebsiella and E. coli species, they also give the same confirmatory test as confirmed by Burgess manual. After the biochemical analysis, there is a confirmation that the coliform is present in our water sample, and now we are going to test that our species sensitive to antibiotic or not. So we have performed antibiotic sensitivity testing. In sensitivity testing, the same 5 isolates like for treated water or untreated water, the same 5 isolates have used, and here is the result that they have shown. Only few species they have shown zone. That means that the species is sensitive to antibiotics, and the results involves are the antibiotic test that we have used. We have used 5 types of antibiotics like amoxicillin, vancomycin, tetracycline, azithromycin, and tobramycin. So the sample is the same like untreated water sample and treated water sample. In untreated water sample, what we have seen that E. coli, it is sensitive to amoxicillin, azithromycin and tobramycin, and the same is the case with the treated water. But the main difference that occurs between treated and untreated is the Klebsiella species. What we have seen that the Klebsiella species is not sensitive to amoxicillin, and the same is the case with tetracycline, and it is sensitive to tetracycline, azithromycin and tobramycin only. But in treated water sample, the Klebsiella species, it is not sensitive to amoxicillin. It is only sensitive to tetracycline and azithromycin, while the Pseudo species is same like amoxicillin, tetracycline and azithromycin, and it is resistant to tobramycin and vancomycin. Sensitivity testing, what we have performed is the cultivation and enumeration of bacteriophages. It involves bacteriophages isolation and polyphages isolation, which is further elaborated by group member Manali Khanna.
After successful biochemical analysis of coliform, we performed cultivation and enumeration of bacteriophages. I would like to tell you about cultivation and enumeration of bacteriophages. As you all know that bacteriophages are bacteria eating viruses. And here, our aim was to make sure phage presence. For this, first of all, phage cultivation was done as we took the both treated as well as untreated sample. And our first step was to make dilution on trypton broth. For this purpose, we made collates from trypton agar, and then we made dilution of trypton broth as we used untreated and treated water sample, and we mixed a little bit of E. coli sample in it for ensure the presence of bacteriophage. And after making the dilution, we add this dilution in our soft agar tubes and pour the soft agar on this spectral plate and give the incubation of 24 hours at 37 degrees centigrade. Now we can see that from results, you can see from this figure, this is the result of bacteriophages, and you can see the visible clear zones, or it indicates the plaque formation. You can see it from graph or a table that for untreated water, we use 10 to the power 5, 6, 7, 8 and 9 dilution. In 10 to the power minus 5 phase dilution, number of PFU was calculated 3, 2, 2, 1 and 1. Well, as you can see from results of treated and untreated water, in treated water, number of phases or number of PFU decreased gradually. Our next step was to cultivate and enumerate number of coliphages, which were phages of E. coli, and you can see from the results that no coliphage was cultivated, so there was no PFU calculated from this experiment. That was all about our group project. Now, I would like to conclude my topic of physical and microbiological characterization of treated and untreated sewage water. So, I would like to call Manal Khalid, which will conclude the topic and will tell you about the importance of our group research project.
I would like to conclude our topic as currently proposed sewage water is an ideal medium for the growth of pathogenic and indicator organisms, and they are harmful for human consumption, yet they can be used for many other purposes. Water quality should be controlled to reduce the risk of waterborne diseases. Careful planning, regular monitoring and appropriate treatments are required for the sustainability of water bodies, animals and plants. So, that's all from our side. Thank you.